No part of this presentation may be copied, reproduced, or redistributed by anyone for any reason. Hello everyone, welcome to Lab 1, Introduction to Immunology. Take a look at the man in this photo. It's pretty gross, right? This is what happens every time you sneeze. Millions of particles are expelled into the air around you. Among those particles are viruses and bacteria. If someone next to you sneezes and doesn't cover his or her mouth, all of those bacteria and virus particles that they spit into the air may find their way into your body when you breathe, and may, in turn, cause you to get sick. While breathing in particles from someone who's sneezing increases the likelihood of spreading disease, it doesn't mean that you will get sick. In fact, on an average day, you breathe in and pick up tens of thousands of bacteria and other potentially infectious agents. So, in other words, your body is constantly bombarded with disease-causing agents such as viruses, fungal spores, parasites, and carcinogens. Despite it all, however, normal active individuals are not constantly sick. So how does this happen? How is it that we're able to stay relatively healthy despite all of the potential disease-causing agents we encounter in our daily lives? It turns out our bodies are actually very well equipped to deal with each of these threats. Our immune system is our body's natural system of defense. Its role in protecting us is fairly complex, so in this lab we will begin by defining immunity and then describe what happens when the immune response is triggered and how this response protects us from disease. The immune system is a complex network of organs, cells, and proteins that work together to protect our body. In studying immunology, we focus on the immune response, the action of those organs, cells, and proteins which together form the body's most powerful and important line of defense against infection. The term immunity refers to any response of the immune system to a foreign substance that enters the body. In other words, immunity is the body's ability to protect itself from disease-causing agents like viruses and bacteria that we encounter on a daily basis. Immunity may be either innate or acquired. The term innate refers to something that is inherent, in other words, something we're born with. Each of us is born with a specific set of tools our body uses to protect us from disease. For example, let's assume that you go out this weekend and eat at Ryan's Steakhouse. A few hours later, you're not feeling too well. You're running a fever and soon develop a nasty case of explosive diarrhea, all because something you ate from the buffet was contaminated with bacteria. And now, you have food poisoning. The fever that develops is a result of your body's innate immune response. By increasing your core body temperature, bacterial growth and replication is impeded, and eventually the temperature will be too warm for those bacteria to survive. A couple of days later, you're back to normal, and you soon forget all about that unpleasant dining experience. So a couple of months later, when you go back to Ryan's, you eat from the buffet and you get sick again, just like before. Unfortunately, your innate immune system doesn't remember that you got sick before. In other words, it lacks memory of the last time you got sick, and so it does the same thing it did the last time. It increases your body temperature, causing a fever. In other words, the innate immune system provides a non-specific response to infection. Each time you get sick from eating the food contaminated with the same bacteria, your innate immune system will respond in the same way. But what happens if you get a cold or the flu instead of getting food poisoning? Again, the innate immune system responds in the same way it did before. In other words, it can't distinguish between the microorganisms that cause food poisoning and those that cause the flu. So whether you're sick from a case of Staphylococcus food poisoning or the swine flu, your innate immune system will respond in the same way, causing you to develop a fever. Fever is one of only several mechanisms of innate immunity. Your intact skin and mucous membranes are extremely important natural ways of protecting your body, as is inflammation of infected tissues. In contrast to innate immunity, acquired immunity develops throughout your lifetime. This occurs through exposure to foreign substances called antigens. An antigen is any foreign particle that enters your body that shouldn't be there. Bacteria, viruses, and parasites are all antigens. Exposure to these antigens may occur as a result of a bacterial or viral infection or through vaccination. Each of you have at some point in your life had vaccinations. There are a variety of different types of vaccines, but the overall effect is the same: to strengthen your immune system. Unlike innate immunity, acquired immunity provides protection against specific pathogens that enter your body. It remembers each of those pathogens so that if they enter the body again in the future, your body can attack them and get rid of them much quicker than the first time around. Acquired immunity is fairly complex, so to understand how it works, we can divide it into two categories: cell-mediated immunity and humoral immunity. Cell-mediated immunity is controlled by special white blood cells called T lymphocytes or T cells. T cells are produced in the bone marrow and, as the name implies, develop in the thymus. There are several types of T cells. Memory T cells remember specific invading antigens. This allows for a quicker response in the future. Helper T cells are extremely important. They are responsible for recruiting B cells, another type of white blood cell, and other T cells to a site of infection. Helper T cells are the cells that are destroyed by the HIV virus. Suppressor T cells inhibit the action of B and T cells, and cytotoxic T cells directly kill invading antigens. The figure to the right shows a T cell as it recognizes and binds to a specific cellular marker in the body. Humoral immunity is acquired immunity mediated or controlled by special proteins called antibodies. Antibodies, like those pictured below, are Y-shaped proteins found in various bodily fluids or humors like blood and urine, hence the term humoral immunity. Antibodies are produced by another type of white blood cell, the B lymphocyte or B cell. To understand humoral immunity, let's look at what happens during a viral infection. Let's assume that while you're studying for your first anatomy test, you get a paper cut while flipping through your notes. Because your skin has been cut, that innate protective barrier has been compromised, and now small particles like the virus seen in the figure can enter your body. Once inside your body, the virus particles begin multiplying. As the virus multiplies, special white blood cells called macrophages recognize the foreign invaders, engulf them, and break them apart. This process is depicted here in the figure. To get a better idea of what exactly is happening, let's blow up this portion of the figure. As you can see, the macrophage disassembles the virus by removing the antigenic particles, which in this figure are depicted as the yellow cones. Once disassembled, the antigenic cones make their way to the surface of the macrophage and protrude from it. So, in this way, macrophages display the foreign antigen on their surface, and when they do so, T cells in the area recognize them. Of the most important T cells that recognize these foreign particles on the macrophage are helper T cells, like the one depicted here. Helper T cells recruit both B cells and other T cells, like cytotoxic T cells, to the site of infection by sending out chemical signals. When B cells receive a signal from helper T cells, they divide into memory B cells and plasma cells. Memory B cells function exactly like memory T cells. They remember the invading antigen so that in the future, if the same antigen enters the body again, the immune system can eliminate it more quickly the second time around. Plasma cells produce antibodies that specifically bind to the antigens on the virus that entered the body. Plasma cells are extremely important because these are the only cells in the body that produce antibodies. Once produced, antibodies enter circulation and bind to the invading virus particles. Macrophages then recognize the antigen-antibody complex engulf it, and break down the virus. This final step prevents the virus from replicating in the body and effectively eliminates it. So to recap, the humoral immune response is quite complex. To simplify it, remember that there are 7 important steps. First, a foreign substance such as a virus or bacterium enters the body where it begins to multiply. Macrophages recognize these foreign particles, engulf them, and break them down, presenting the antigens to nearby helper T cells. The helper T cells then send out chemical signals to other T cells as well as B cells, recruiting them to the site of infection. Once there, B cells split to form memory B cells and plasma cells. Plasma cells produce antibodies that bind specifically to the invading particles. Macrophages then engulf the antigen-antibody complex and break it down, destroying the foreign particle. That concludes this podcast, Introduction to the Immune System. To help you better understand the processes involved in the immune response, follow the directions outlined in the lab handout.